second objective here, we're getting into some of the physical and structural supporting structures in the central nervous system. Now, most of this should be a review in terms of things we've spoken about already in the lab. And so we'll kind of go through this, we'll kind of get into this here and then pick it up the next time we meet. But the first set of structures we want to think about as supporting and protecting really the central nervous system is the cranium. So the bones of the skull, the bones of the vertebral column, which we talked at length about, which help to house and protect the brain and spinal cord, are probably the first line of protection of these structures. We've also got the meninges. So we've got the dura mater, the arachnoid mater, the pia mater, and these are all playing a protective role in that not only are they covering these structures physically, but then they also allow for the suspension of cerebrospinal fluid. So cerebrospinal fluid exists within the arachnoid and pia mater, so in the subarachnoid space, and that cerebrospinal fluid allows these structures to be suspended, to be kind of weightless in these cavities, in these structures. And I talked about this in lab, but if you think about the brain being this very delicate tissue, we don't want it to be sloshing around in the cranial cavity because it's really hard, it's dense bone. And so having the meninges to cover these structures, but also having the CSF to kind of suspend these structures is also a part of that support and protection. Okay, so here's just a schematic or an illustration of these structures kind of working together to protect the brain and spinal cord. We've got the hard cranium or the skull, which is the outer dense bone that forms the cranial cavity. We've got the dura mater, that tough meningeal layer, We've got the arachnoid mater, that kind of mesh-like layer, and then the pia mater, which is one with the brain. And so in between these two layers, the pia mater and arachnoid mater, is where we have the circulation of that cerebrospinal fluid all around the brain and all around the spinal cord. Okay, if you think about the vertebral column, it is also playing a protective role of our spinal cord as well. Here we can see a look at those ventricular spaces. So we talked a little bit about the cavities in the brain where we've got the subarachnoid space where we have that CSF kind of floating around these structures. And so this is just another view of that where we can see those that CSF being circulated here around brain as well as spinal cord. But then if we look at these areas of the brain where we have these arachnoid granulations or arachnoid villi, so these outward projections that we're seeing here into this space, the space is called a sinus. This is how we can reabsorb that CSF. So CSF is not just floating around indefinitely, it's being secreted by the ependymal cells of the choroid plexus, and then it is being reabsorbed by the arachnoid granulations, these openings of the sinus here, and these outward projections, which are called arachnoid villi, that will then reabsorb that CSF, and then fresh CSF can be secreted. And so we're always renewing, refreshing the CSF that is being circulated. All right. So we're now moving on to look at the structure or the makeup of CSF and how it circulates in those ventricles, those cerebral ventricles, as well as what the electrolyte and fluid contribution is to that fluid itself, that clear, watery cerebrospinal fluid. So we said that CSF is made by the ependymal cells. They're one of our glial or supporting cells that are found lining the choroid plexus right around our ventricles. And then it's going to circulate through those ventricular spaces as well as the subarachnoid space, and then be reabsorbed by the arachnoid villi that I just pointed out. And its function is to cushion the brain as well as maintain a stable interstitial environment in terms of the makeup of the interstitial fluid that is around the brain. And again, this is really to set it apart from what's happening in the blood. We want a separate interstitial fluid environment in the brain so that if there are any toxic exposures, any viruses, pathogens in the blood, those are separate from what's happening in the fluid that is surrounding the nervous tissue. Here is the makeup of plasma and comparing that to the makeup of cerebrospinal fluid. And while I don't want you to memorize these numbers here, I do want you to think about the trend. So the idea that there is no plasma, right? Comparatively, there's very little protein in our cerebrospinal fluid when compared to the plasma. Everything else is kind of pretty much the same. So there is less glucose, but then much less proteins when we compare plasma to CSF. And that's kind of the trend that I would prefer you pick up from this than actually memorizing these values. So we'll look at the structure of the blood-brain barrier, but because of the fact that there's that separate environment, glucose cannot easily move into the brain and proteins cannot easily move into the brain because of that barrier that is, being formed between the blood and what's happening in the interstitial fluid and the cerebrospinal fluid. Okay. This is just looking at those ventricular spaces, which we should already be familiar with at this point. Our lateral ventricles communicating to our third ventricle here, communicating to our fourth ventricle, and then eventually going from fourth ventricle all the way around our spinal cord in the subarachnoid space, and then eventually being reabsorbed in these sinuses here. So here's that sinus that we looked at. Here are those arachnoid granulations, or arachnoid villi is another way to another word for that term. And these are the structures that are reabsorbing that cerebrospinal fluid and keeping that circulation